Sixth-year biology students sitting your exam in June 2026. Let's talk about everything that you need to know for diffusion and osmosis. So diffusion and osmosis is a Unit 2 chapter, and don't forget, question 9 mandatory experiment belongs to this chapter as well. So we're going to talk about diffusion first of all. Diffusion is the movement of particles from an area of high concentration to an area of low concentration. Now, it is described as passive transport because those particles do not require any external energy to be supplied to them. Because if we have a look here for a second, imagine we had all of these particles together. And a typical example was if I got a perfume bottle, okay, or if I have a whiteboard spray. If I get a whiteboard spray here, there's a high concentration of whiteboard spray where I am going to spray it. High concentration. What's going to happen when I spray it is eventually the particles, using their own random kinetic energy, are going to move. And they're going to move to occupy and spread out into the entire container that they're in. And no external energy is required for that. So examples of this would be perfume, or we can have diffusion in gases or liquids. So for example, food coloring diffuses from high concentration to low concentration. No energy, external energy is required. Particles randomly move. Okay, so that's diffusion. Now, osmosis is a special type of diffusion because we are going to be concerned with the movement of water molecules only, and we're concerned with a selectively permeable membrane. So the definition for osmosis is the movement of water molecules across a selectively permeable membrane from a region of high water concentration to a region of low water concentration. Now, to understand this better, we need to understand words like solvent, solute, and solution. The typical solvent that we talk about in this chapter is water, and the proportion of water to our solute, like our salt or our sugar, will determine the concentration of a solution. Imagine I get a plant and I'm going to water the plant. So I put all of this water into the soil. Now the soil has high water concentration than the plant cells themselves. Water is going to move from the soil in through the selectively permeable cell membrane into the lower water concentration in the cells themselves, because that's what osmosis is. Selectively permeable membrane allows certain substances to pass through, smaller particles like oxygen or water or amino acids. Doesn't allow larger molecules like starch or proteins to pass through. Okay, so that's selectively permeable. Cell membranes, the nuclear membrane, or you maybe use visquing, tubing, or cellophane for your mandatory experiments. So when we talk about osmosis, we need to know osmosis in a plant cell and osmosis in an animal cell. But the factors, I suppose, that affect the rate of diffusion and therefore the rate of osmosis are the following. Concentration gradient. The higher the difference between the concentrations inside and outside the cell, the quicker the rate of diffusion. Because the greater the difference, the particles are going to move quicker. Okay, so that's concentration gradient. The next one is temperature. If I supply energy, temperature, heat, if I supply energy in the form of heat to the particles, what's going to happen is those particles are going to vibrate faster and then they're going to move faster. So that's what higher temperatures supply more energy for particles to move, giving a faster rate of diffusion. And another factor that affects the rate of diffusion is surface area. So the larger the surface area, the more particles can go around that surface area and the faster diffusion can take place. So surface area also affects the rate of diffusion. Now, if we talk about osmosis in an animal cell, we're going to use a red blood cell as an example. And remember, a red blood cell, side note, is the only animal cell at maturity that doesn't have a nucleus or mitochondria. So we can have three different types of beakers. We can have a beaker with our red blood cell that is equal concentration. So inside and outside the cell are of equal concentration. In that case, we've created an isotonic solution. Water moves freely in and out of the cell at the same rate. However, if outside of the cell is high water and inside of the cell is low water, now I've created a hypotonic solution. Water moves from high water outside the beaker into the cell, low water concentration, and the cell is going to swell and burst. And we have a hypertonic solution where we place our red blood cells in low water concentration. So inside the red blood cell is high water, outside is low water. Water will leave the red blood cell and it will shrivel, and that is known as crenation. Now if we compare that to our plant cell, remember plants have a vacuole and they also have a cell wall. So what's going to happen again in an isotonic solution? You put a plant cell in a beaker of water that's of equal concentration to its cytoplasm. Water moves freely in and out at the same rate. However, if you place a plant cell in a beaker of high water concentration, water rushes into the cell, the vacuole collects the water, the cell swells, the cell membrane pushes against the cell wall, and that creates turgor pressure, which plants rely on for their strength. And then that would be our hypotonic solution. And the last one then is our hypertonic solution. If you place a plant cell in a beaker that has low water, high solute, water will leave the cell, and what's going to happen is the cell membrane detaches from the cell wall, and that's what we call plasmolysis. To reverse that, or wilting, we would put a plant in high water. So that is what you need to know for diffusion and osmosis in a plant cell and in an animal cell. Remember, diffusion is passive transport because the molecules move using their own kinetic energy. Osmosis is a special type of diffusion because it's concerning water molecules only, and it must be across a selectively permeable membrane. An advantage to a cell of having a selectively permeable membrane is the control. So what substances enter and exit. It can keep harmful substances or harmful organisms, even like pathogens, out. So make sure you know how to draw and how to describe these for Unit 2, diffusion and osmosis, and always ask yourself, where is the high water concentration?